Hello, everyone, and welcome to my presentation. I'm doing a presentation on a paper called Behavioral Correlations and Aggression in Praying Mantids, done by Fisher, Howell, and Price, which was featured in Behavioral Ecology and Sociobiology 2020. My name's Katie Justison. So before I start, I wanted to do a little presentation layout. The first thing I'm going to be talking about is the introduction, which I'll talk about the species as well as precopulatory cannibalism in general. And then I'm going to go into hypotheses on why precopulatory cannibalism exists. Then furthermore, I'm going to talk about the study aim as well as the methods, the results, and the discussion. Then fifth, I'm going to do a critical evaluation of the results. So, Mantodea. Mantids belong to the order of Mantodea, which is characteristically defined by their triangular head and raptorial legs. As you can see, mantid raptorial legs are strong, spiky legs used for holding and grasping their prey. Additionally, these legs are used in copulation to hold their partner correctly in order to mate. The most diverse feature between Mantodea is its food niche. Most research suggests that mantids are predatory, cannibalistic arthropods, which can cause major food limitations and therefore energy limitations for these creatures. This lack of access to resources then causes issues of reproductive success, which may lead to significant impacts, such as copulatory cannibalism, which I'm focusing on today. Most mantids are sexually dimorphic, meaning that there is a male and female sex within the species. Most mantid life cycles are similar. However, it is important to note that not all are the same. The species is mainly univoltine and semiparous, meaning it undergoes a complete life cycle and produces one brood per growing season without requiring diapause. Oviposition, the laying of eggs, occur in the fall and then overwinter until they hatch in the spring. As shown from the figure, hemimetabolous insects go through an egg, various nymphal stages, and then the full adult stage, as they do not undergo complete metamorphosis. Each nymphal stage is characterized by the term instar. Mantids, as well as all other hemimetabolous insects, release molting hormones in order to separate and shed their existing cuticle so that they can have more surface area for growth and water retention. The main goal of the nymph is to be an effective processor of food in order for growth efficiency to adulthood, whereas the adult's main goal is to mate, which is important in this study. Male mantids typically choose females to mate with based off fecundity. This means they will choose a large, more fecund female over a small one. However, we know that females typically have choice in environmental situations due to their larger, more invested gametes. Females hold out for males that seem to have superior genetic quality or can provide direct benefit availability of resources. This conflict can result in sexual cannibalism. Precopulatory cannibalism can be found among many invertebrate species, such as arachnids and insects, including mantids. Precopulatory cannibalism is the consumption of potential mating partners before copulation, which can leave females unmated if males are rare. This is predicted to have high correlation to high aggression towards prey. Precopulatory cannibalism could reduce population growth and increase population extinction, as well as can be maladaptive for both sexes, as females increase their risk of dying without reproduction. However, there are benefits of the nutrients or potential paternity assurance in cannibalism. So why does precopulatory cannibalism exist? The first hypothesis for why precopulatory cannibalism exists is because females are able to gain essential nutrients for egg production by eating their mate. This hypothesis is especially popular when resources are scarce. Hypothesis 2, mate choice. The second hypothesis is that precopulatory cannibalism could reflect an extreme form of female mate choice, as non-preferred mates, those who are less genetically fit, are devoured to prevent copulation. Hypothesis 3, aggression spillover. The third hypothesis and most prevalent is the aggression spillover hypothesis. This hypothesis suggests that precopulatory cannibalism can be costly to females. However, this cost is counteracted by the benefits of high juvenile feeding rates. If this is the case, then there are two predictions. The first is that prey and cannibalism aggression is genetically correlated within a species. The second is that male aggression towards prey may be less due to constraints of potential costly behavior, as females must be more aggressive. The aim of this study is to examine the aggression in context of praying mantids residing in New Zealand and how that relates to cannibalistic behavior. The study used two species of praying mantis, commonly known as the springbok mantid and the New Zealand mantid. The springbok mantid is known to be highly aggressive, whereas the New Zealand mantid is known to have low aggressive behavior. Hello, and welcome to my small presentation within a presentation on the method section of this article. The study had four main predictions. The first was that males were going to be less aggressive than females for all species. The second is that springbok mantids will be more aggressive than New Zealand mantids. The third prediction is that springbok mantid aggression would be linked with their life cycle stages. And the fourth study prediction is that springbok mantid female aggression would be linked with mating. So let's get into the 5 W's of how they did this experiment. Fisher et al. Decided to do a study testing aggression across 2 species of mantid in different life stages. Fisher and Price both attend the University of Liverpool, and Howell studies at the University of Auckland. The study is important on understanding aggression and how it's linked to cannibalism behavior. They had 2 sampling periods, the first in February and March of 2017, and the second in February 2019. To ensure mantids were virgins, they collected only juveniles. New Zealand mantids were collected in grass and woodlands outside of Auckland, and springboks were collected in suburban gardens inside of Auckland. Okay, so how'd they do the experiment? In order to qualify aggression, the mantids were starved for 2 days prior to testing. They were also weighed in order to compare different instar stages in different time periods. They were then placed in a box where a virtual prey item was created and projected. Mantid aggression was determined by whether or not they attacked the virtual prey item. After the virtual trials, mantids were given a fly and 10 minutes to catch it. If they succeeded, the process was continued until either they hit max trials or stopped catching the fly. Females were starved for a duration of 2 days. Then males were introduced to the female cage to test the aggression towards potential mates. This was done at night because it is known that cannibalistic activity occurs prominently after dusk. Males and females were left together for 12 hours, and it was recorded whether or not the male was cannibalized or mated. This was done once per individual. Generalized mixed models, binomial error correlation, 4-way interactions, Gaussian error, Spearman rank correlation, and behavior repeatability were all used in the statistical analysis of the results. Alrighty, now let's go see what they found. Going back to the 4 predictions mentioned earlier, the first suggested that female springbok mantids were found to be more aggressive than prey. This was found to be significant through the generalized mixed models and Spearman correlation comparing springboks to New Zealand mantids. Using generalized linear mixed models with binomial error correlation, individuals who were tested whether they attacked the prey item taking into account their age, sex, or species. These results came back significant as females were found to be generally more aggressive than males. So this is a graph that shows the mean probability of attacking a virtual prey item, as indicated by the red box, as well as looking at the difference in the y-axis. This shows that female springbok mantids were more aggressive than New Zealand mantids, which proves prediction 1. Female springboks have a lower body mass and are flightless. Instead of worrying about dispersal capacity, the female springboks are selected to be more aggressive in order to maintain a high feeding rate that could potentially increase egg production and fitness. Females would benefit more from their own survival, prolonging reproduction in environments with low resources, as shown by this data. Therefore, females choose the benefit of gaining nutrients over the cost of losing a mate. This also shows that females are more aggressive than males in general, which proves prediction 2. Fisher et al. Suggests that males may be less prone to cannibalistic activity as their sex disperses in the population. Males feed less during adulthood in order to maintain their weight for easier flight dispersals, as they may keep up with the energy demands of disseminating. The results showed that adult females were significantly heavier than adult males in both species, with both the results having a low p-value below 0.001. This further indicates that aggressive behavior is sex-dependent, as females continuously show that they are more aggressive than their male counterparts due to dispersal factors. So for the third prediction, through Spearman rank correlation in generalized linear mixed models with Gaussian error, the results suggest that springbok aggression is linked to different life stages, specifically in males. And you can see that this is significant due to the p-value. However, the data and results suggesting springbok female aggression linked to mating was found to be insignificant, as also shown by the p-value. For female springbok mantids, their attack rate did not change due to life cycle stages, as shown in graphs A and B, which could explain why prediction 4 was found not significant. There was also little variation in difference of aggressive behavior between juvenile and adult New Zealand mantids, compared to springbok mantids, where there was a huge difference. This shows that springbok mantid aggressive behavior depends on life stages and therefore significantly correlates with prediction 3. Fisher et al. Suggest that aggressive behavior in juveniles is fairly equal between sexes, as they both have the main goal to eat and gain as much energy as possible before reaching full maturation. However, once the adult stage is reached, the priority of energy investment is decreased as the priority of mating is increased, specifically in males, which may be the result of why males decrease aggression in adulthood. How do the results link to cannibalistic behavior? Well, out of the 12 trials done with the springbok species, 5 were cannibalized. This is compared to the 7 trials done with the New Zealand species and 1 cannibalized. The study found that precopulatory cannibalism may not be an indication of aggressive behavior across mantids. Instead, it may be correlated with individual need. Previous studies have suggested that cannibalism rates occur and are determined by environmental cues rather than behavior. Examples of these environmental cues could be low food resource or a low operational sex ratio, meaning low male availability. The results of the study shows that high rates of precopulatory cannibalism may not be because of the foreseen hypothesis of aggression spillover. Instead, the data indicates that sexual cannibalism may be species dependent. With the results present, I suggest that the most likely cause for cannibalistic behavior parallels with the foraging hypothesis outlined in the introduction. This is because females are more likely to cannibalize when resources are scarce, and therefore they benefit from obtaining essential nutrients for further survival. The results also showed that the benefits of aggression for females may outweigh the costs of aggression, as prolonging their own adult life is more beneficial than losing a current mate and trying to reproduce in an environment with low food availability. However, a critical evaluation of the results is that aggressive behavior seems to be species dependent, as aggression of springbok mantids were significantly higher than that of New Zealand mantids through all trials. As well, aggression behavior may also be environment dependent, as the data suggests that aggression could evolve with variation of different environmental cues, such as mating availability. There also seems to be some limitations within the study. To recall, the study had 2 different size groups of mantid species, which could skew the data, as they were comparing percentages of different sized populations. Increasing the sample size could better solidify the study's findings. Additionally, testing different species of mantids could further benefit the understanding of aggressive behavior within the taxon, which may warrant further studies. Alrighty,